Hi, I am Aurel Enriquez, and this presentation contains our discussion on cell structures and their functions. Cells have components that could be classified into these three, right? So it could be either classified as an organelle, the cytoplasm, or the cell membrane. So yung mga organelles po, these are the tiny, different specialized structures, and each of them have different functions. So listed here are examples. Okay, so iisa-isa hindi naman po natin to as we proceed with our discussion. Next is the cytoplasm. So the cytoplasm is a fluid or jelly-like substance that holds the organelles. So they look like they're just floating on the inside of the cell. Okay, so dito pa lang po, we could already have an idea that our cells are in fact approximately 60 to 80% water, 60 to 80% fluid kasi isang cell pa lang po contains that much fluid, or as it says here, yung jelly-like substance to fill up one cell and hold all the other structures or organelles inside. Okay? Next is the cell membrane. So, pwede na rin natin itong tawagin na plasma membrane. So, this encloses the cytoplasm together with all the organelles inside of it. So, kumbaga yung cell membrane po, yun yung container or lalagyan ng mga organelles and the cytoplasm that holds those organelles. This slide shows us the generalized structure of a cell. Now, as we progress through this semester, makikita natin na may mga iba't ibang cells within our body na hindi rin naman parang exactly the same yung structure dito sa nakikita natin dito. Pero ayun nga po, most of the cells in our body have a similar structure such as these. And of course, as we go through this discussion, titignan din po natin isa-isa yung mga structures na nakikita natin dito or yung function ng bawat isang organelles that we're seeing in this illustration. Cells are known as the smallest units of life. So if you would remember from previous discussions, before human organs would actually become organs and before they would be tissues, to create these organs, we need a bunch of similar cells that are put together in a way that makes functional cells. So without these small units, without these cells, we would not have living organs. Sa mga cells din po nagaganap yung metabolism and energy use. Even energy production, that also happens within each individual cells. So may capability yung bawat isang cell to create the molecules necessary to produce energy and use that energy for whatever purpose they may serve. Next is communication. So cells are also capable of communicating with one another. All right, cells are capable of communicating with one another through many different ways. So either magpo-produce sila ng enzymes na makaka-apekto sa kalapit nilang cell. They could produce hormones that could give instructions to other cells or organs that are further away from them. So when cells are being damaged, yung products naman po ng cell death could also send a message to cells, to our immune system, to check why cell destruction is happening in a specific area. So basically, kaya rin po mag-usap-usap ng mga cells natin by sending each other chemical signals. And of course, cells are responsible for reproduction and inheritance. So as one cell matures, it would eventually have to make a new copy of itself through cell division. And of course, this new cell would also have a copy of the genetic material or yung DNA ng original cell na pinanggalingan niya. So that is a simplified idea of how inheritance could happen. Let's now talk more about the cell membrane. As I have oversimplified earlier, this is the container kung saan nakalagay sa loob lahat ng mga organelles and cytoplasm. So the materials inside of the cell are referred to as intracellular components. All right, ulitin po natin. Things that are inside of the cell are referred to as intracellular components, and those that are outside are called extracellular components. Okay, so yung mga bagay na nasa loob ng cell, intracellular. Lahat ng iba pong bagay na nasa labas ng cell, extracellular. So the cell membrane also acts as a selective barrier. So ang cell membrane po, it actually has these tiny pores or channels that could open up whenever it is needed. Pero hindi po ito basta-basta nag-open up to any material that attempts to get inside of it. So that's what we mean when we say selective. All right. So imagine our school campus as the cell. Tapos we have gates where not everyone could get in. So ayan, kailangan muna ng ID para makapasok. So that's kind of like the idea of how the cells would produce kind of like a selective characteristic. Still talking about the cell membrane, they have what is known as a phospholipid structure. So this thing actually has two layers, and each layer are facing different directions. So on the next slide, my picture tayo for reference, but basically one phospholipid molecule has a hydrophilic head, meaning that yung specific portion na yun ng phospholipid is preferred to be in contact with water, or is okay to be in contact with water, either those that could be found on the outside of the cell or to the inside of the cell. So sa kabila naman po, it has a hydrophilic tail, meaning that its usual positioning is away from any water source. This photo right here illustrates what we just said. Yung mga blue na circles po, no, those are the hydrophilic head of the phospholipid molecule. So it's either facing the outside of the cell and also the inside that contains the cytoplasm, or again, the jelly-like material. And also there's this layer in the middle, right? So basically nakaharap sila sa mga areas kung saan natatakpan sila, no, from the water source. So these ones in the middle, we would refer to these as the hydrophobic tails. So notice na dito lang sila nakaposition sa areas wherein they don't make direct contact with water, because again, they are hydrophobic. All right, so again, the hydrophilic heads, all right, are facing either the outside or inside of the cell, because those are the areas where there's water or fluids, and the hydrophobic tails, they are found in the middle of that double layer structure, because they are attempting to avoid water exposure. On this next slide, we've already talked about this earlier. The cell membrane is selective, all right? So it kind of has like the idea of no ID, no entry. So the actual term for this one is selective permeability. So let's look at the important statements here. Same with all my other discussions, naka-highlight na po the important parts. So this one allows only certain substances to pass. Right, so substances such as enzymes, glycogen, and potassium are found in higher concentrations inside of the cell. So sa loob daw po ng cell, pwede nating i-expect na mas maraming enzymes, mas maraming glycogen, which if you would remember from the past discussion, is usually a storage form of energy, or a carbohydrate storage form of energy, and potassium, which is a type of electrolyte. And the next substances such as sodium, calcium, chloride, these ones are in higher concentrations on the outside of the cell. So yung space sa labas, yung extracellular fluids, no? Those are the areas wherein we would most likely find these types of salts. So sodium, calcium, and chloride. On this slide, let's focus on, again, just the important parts. It says here that substances like oxygen and carbon dioxide can easily pass through, or could pass directly through the cell membrane's phospholipid bilayer, which is a very important task considering that a lot of chemical reactions require oxygen. So tama lang na madali lang maka-enter yung oxygen to the inside of the cell, because it's very important. It's needed for a lot of things. And also it makes sense, no, na pati yung carbon dioxide madali lang makadaan papalabas ng cell because carbon dioxide is a waste product. All right, so excessive amounts of carbon dioxide accumulated inside of the cell could be harmful to the cell. So again, it makes sense that both oxygen and carbon dioxide could easily pass through, could enter and exit the cell directly. Next, there are some substances that must pass through transmembrane protein channels such as sodium. Alright, so kumbaga ito po is may specialized structure pa kung saan pwedeng dumaan or kung saan dapat dadaan ang sodium. This slide talks about other substances and how they get inside of the cell. Next is glucose. So if you remember from past discussions, glucose is a monosaccharide, and this is also the most basic structure of sugar or basic structure of carbohydrate that could be utilized by our cells for energy production. So for glucose, this one, although it is a very important substance, hindi rin ito basta-basta nakakadaan through the cell membrane. So it needs carrier molecules, meaning it needs other structures, no, or molecules na dadalhin ito papunta sa loob ng cell. It can't just pass through the cell like oxygen and carbon dioxide could do. Next is some substances require a vesicular transport across the cell membrane. But what does that mean? So there must be a vesicle, right, or kind of like a container, no, a sac, right, or kind of like a bag that must fuse with the cell membrane for transport. So ano pong meaning na? Yung vesicle, it would have to kind of like connect or yun nga, fuse with the cell membrane or become one with the cell membrane to release the materials inside of it. So those materials inside of the vesicle would now be inside of the cell membrane. So that's what I mean when we say vesicular transport. Next, let's talk about active and passive transport. When we say passive transport, this does not require energy, all right? So, hindi gagastos ng ATP molecule yung cell to proceed with transporting these certain substances. But when we say active transport, they require the cell to expend energy, usually in the form of ATP. So, gagamit or gagastos ng ATP molecule ang isang cell para lang po mapadaan yung mga substances na ito. Listed here are the different types of passive and active transport. For passive, we have what is known as diffusion. Next is osmosis, and we also have what is known as facilitated diffusion. Now, for active membrane transport, magpo-focus po tayo sa endocytosis and exocytosis. Alright, ulitin po natin, there are a few other methods of active transport listed here, but for this discussion we would be focusing on endocytosis and exocytosis. Let's first talk about the different types of passive transport. First up is diffusion. So again, let's focus on the important statements here. Diffusion generally involves the movement of substances in a solution down a concentration gradient. So we've used quite a few terms in here, so i-define muna po natin yung mga terms na nagamit. First, a solution is generally composed of two major parts, all right? So the solutes and the solvent. So yung solution po, basically this is a mixture of two different things, all right? Next, the solutes, these are the substances that are being dissolved in a liquid known as the solvent. Alright, so yung solutes, yun daw po yung mga particles, yun yung mga electrolytes, yun yung mga materials na usually either nadidissolve sila or what term do we use? Kumbaga is parang nalilesen yung amount nila or nadadilute sila once they are being exposed to the solvent. So the solvent is any type of fluid that has the capability to dilute materials or dissolve those materials, which again are referred to as solutes. Right, so solutes, ayan po, they tend to move from an area of higher concentration to an area of lower concentration. So usually daw po in the process of diffusion, when solutes are gathered in one specific area, the tendency is that they would like to create balance. So when they are placed in an environment kung saan mas kaunti sila sa labas, this would encourage them to move to the outside. Right? So again, from an area of higher concentration, which is inside, they would move to an area of lower concentration, which is outside. Now, that would eventually even out the amount of solutes, no, both on the inside and outside. All right, so this movement from high concentration to low concentration is now referred to as diffusion. This slide further discusses to us what a concentration gradient is. So the concentration gradient is the difference in concentration of a solute in a solvent between 2 points. So as I've said in the previous slide, for easier understanding, no, so gamitin na lang muna natin yung terms na inside and outside. So in this case, maybe it's like the inside of the cell and the outside environment. So pag sinabi concentration gradient, there is a difference in the amount of solutes or particles dun sa inside ng cell in comparison to the outside of the cell. This illustration explains to us how diffusion happens using a salt cube, all right, a salt cube that has been placed inside a beaker of distilled water. So as usual, or as you would know, no, salt cubes, they're concentrated. No, they're a concentrated structure of salt that has been compressed. And again, they don't have any other components aside from the salt particles, sodium and fluoride. So if we place it in a beaker that contains this much amount of water, naturally, no, the distilled water does not have any salt content. So following the idea of higher concentration to lower concentration, the salt particles would start to move out or move away from the salt cube or yung area kung saan marami sila. And then they would start moving into the environment or surrounding environment, which is composed of water. And again, distilled water does not contain any salt particles. So what would happen eventually is that this would kind of like even out the amount of salt everywhere else. As we can see right here, na-dissolve na po yung salt cube. The salt cube no longer exists. It's just a liquid, no? It's just a solution that has an even amount of salt all throughout. So that's how diffusion happens. Starting from an area where there is a very concentrated amount of particles, we would end up with an area that is, the solute is now evenly distributed within that mixture. So again, that is how diffusion would happen. Next is osmosis. So osmosis is the diffusion of water across a selectively permeable membrane from a region of higher concentration to lower concentration. So the idea is kind of similar, pero imbis na yung mga particles yung kumikilos para ma-achieve yung even distribution, yung water po yung mag-move from one area to another to achieve the even distribution. So when this happens, osmosis would exert a pressure, otherwise referred to as osmotic pressure, which is the force required to prevent movement of water across the cell membrane. We would better understand this as we look into the next slide for an illustration. This illustration right here shows us how osmosis would happen. All right, so in this example, again, we have a beaker that is filled with water, and then we have this flask right here, or kind of like this inverted funnel right here kung saan there is a salt solution on the inside, but there is kind of like a cloth covering on the outside or on the opening on this segment or in this area right here. So what would happen is, because of this cloth, once we submerge the container into the tube that contains distilled water, since the membrane is selectively permeable, hindi kasha yung mga salt particles na nandun sa loob ng container. So as you can see right here, there are very large salt particles that look like green spears, and then there are these medium-sized salt particles that are represented by the pink spears. All right, so as we can see, hindi sila kasha dun sa holes ng selectively permeable membrane. But what else are we noticing here in this illustration? the holes of the selectively permeable membrane, kasya doon yung particles ng water, right? The water molecules, sorry, not particles, the water molecules, they could pass through the selectively permeable membrane. So they could enter the container, pero yung mga salt particles on the inside, they can't exit the container. So what is happening here? Since hindi makamove palabas yung mga salt particles, yung water na mismo yung magmumove to the inside of the container. So that is how osmosis happens. No? Instead of the particles moving, the water is the one that would be moving from a higher concentration to lower concentration. In this case, mas mataas yung concentration ng pure distilled water. Dito sa labas, mas low yung concentration ng water dito sa loob ng container. So now, eventually, they would achieve some form of balance kung saan, ayan, equaling amount ng water on the outside and on the inside of this container kung saan natatakpan yung kabilang end ng selectively permeable membrane. Alright, so ayun po, that's the general idea of how osmosis would happen. It's kind of the same concept of moving from a higher concentration gradient to a lower concentration area. Pero ayun nga, in this time, instead of the particles moving, it's the water that is moving. In this slide, let's now talk about the effects of osmotic pressure on cells. All right, so yung movement ng water from one place to another, again, that creates pressure, and that could also create effects on a cell that we may be able to observe. So ayun po, the cell may be placed in solutions that could either be hypotonic, isotonic, and hypertonic. So on the next slides, let's look at what these terms would mean and anong kayang gawin ng mga ito doon sa isang cell. First up, let's talk about a hypotonic solution. So let's look at this statement right here and try to understand it piece by piece. A hypotonic solution has a lower concentration of solutes, so mas konti lang daw yung particles dito sa solution na to, and a higher concentration of water in comparison to the inside of the cell or to the cytoplasm of the cell. Okay, ulitin po natin kapag sinabing hypotonic, ang solution, alright, konti lang yung solutes or yung particles, mas maraming water dun sa solution. So let's look at the effects. Water would move by osmosis into the cell. So mas mataas daw yung amount of water on the outside, so it would go to the inside, causing the cells to swell or enlarge in size. Alright? Now, if the cell would swell enough, alright, to the point na hindi na nito kayang dalhin yung kind of like excessive amounts of water, it can now rupture in a process that is referred to as lysis. Next is an isotonic solution. So a cell immersed in an isotonic solution has the same solute concentration on the inside and outside of the cell. So same lang daw po yung amount of particles sa labas or dun sa mismong solution, and same lang din yung amount ng particles dun sa inside ng cell. So in a way, there's equal amounts of everything both on the inside and outside of the cell. So this will basically do nothing. All right, so the cell will neither shrink nor swell. It would not affect the cell because it's kind of like a very stable environment. Next are hypertonic solutions. So hypertonic solutions, again, let's look at the statement right here, piece by piece. So the cytoplasm of a cell in a hypertonic solution has lower solute concentration. Mas kaunti daw yung particles or yung solutes no, sa loob ng cell. Mas maraming particles on the outside of the cell or at the environment. And a higher water concentration compared to the surrounding solution. Alright? So, mas mataas daw yung water content sa loob ng cell kesa sa labas. Alright? Again, let's simplify that. When we say hypertonic solution, more particles are on the outside, less particles are on the inside. Pag sinabing hypertonic, less water is on the outside, more water is on the inside. Okay? So what would happen? Water would move by osmosis from the inside of the cell to the hypertonic solution. So yung water daw po, lalabas siya dun sa cell kasi marami or high yung concentration gradient dun sa loob ng cell, and it would move to the outside. Now, the loss of water volume would lead to cell shrinkage or crenation. So, parang magmumukhang liliit or gugusot yung cell kasi nga nabawasan yung contents nito dahil nag-move from the inside of the cell to the outside yung water content. So, that's what happens if we place a cell in a hypertonic solution. This slide shows us the effects on a cell, specifically a red blood cell, if we move them through different solutions of different concentrations. Again, when we place it in a hypotonic solution, since mas maraming water on the outside, this would move to the inside of the cell, causing the cell to enlarge or swell. Eventually, kapag masyadong water or masyadong maraming water na yung makakapasok dun sa cell, the cell would burst. Next, if we place a red blood cell in an isotonic solution, again, there's equal amounts of everything everywhere. So nothing would happen kasi stabilized yung environment. Next is if we place a red blood cell in a hypertonic solution. Again, mas maraming particles dito sa labas, mas maraming water dito sa loob ng cell. Now, what would happen is the water would move to the outside of the cell, and that loss of water would lead to the cell shrinking and yun nga, looking like it's wrinkled or crenated. Okay, so that's the negative effects of placing a cell in either a hypotonic or hypertonic solution. Let's now talk about facilitated diffusion. So when we talk about facilitated diffusion, lipid-soluble substances such as oxygen, carbon dioxide, they could diffuse directly through the phospholipid bilayer. Next, let's compare that with water-soluble substances. Kung water-soluble substances naman yung mga dadaan, examples of this would be ions, they could only pass through the cell membrane if they would go through certain channels or if they are being carried by carrier molecules. All right, ulitin po natin, lipid-soluble substances like oxygen and carbon dioxide, they could pass through freely. But if a substance is water soluble, then they would have to go through a certain channel or a certain structure on the surface of the cell, or a molecule has to bring them to the inside of the cell. This illustration shows us how the different ways of diffusion could happen. So as we can see right here, lipid-soluble molecules could pass through easily without any problem. Now, if we were to look at this illustration right here, there are non-lipid soluble molecules, all right, or again, in the previous slide, they have been referred to as water-soluble molecules na hindi makakadaan basta-basta. So they would have to go through membrane channels or specialized structures within the phospholipid membrane that would serve as kind of like a passageway for them to get through, which again, hindi ito magiging posible kung wala ito at yung mismong bilayer membrane lang yun na dyan. Alright, so again, that's what we mean when we say facilitated diffusion. Their passage is being facilitated or assisted by a different structure. This next slide talks about leaked and gated channels. So these ones are quite easy to understand. When we say leak channels, they constantly allow the passage of ions, right? So walang restriction, no? As long as the ions are in the correct area and they're in the correct passageway, they could easily get in and out of the cell. Comparing that to gated channels, they limit the movement of ions across the membrane by opening and closing. So it's literally like a gate kung saan na nakokontrol yung amount of ions passing through by opening and closing certain segments of the channel. This is an illustration of leaked and gated channels. As we can see right here, for leaked channels, diba, it shows here that there are potassium ions that could pass through. So it's always open. Potassium ions have no trouble getting into the cell if they pass through these leak channels. Now, in comparison to sodium has to go through these gated channels kung saan nare-regulate yung amount nila on the inside of the cell kasi nga hindi all the time open yung mouths or yung kumbaga yung entrance ng gated channels. So again, that's a side-by-side comparison between the leak channels for potassium and the gated channels for sodium. Next, let's talk about carrier molecules. So carrier molecules, these are made up of proteins that assist with facilitated diffusion. So carrier molecules, they could move the water-soluble molecules or ions across the membrane. Alright, so literally kaya nilang i-carry or kaya nilang dalhin yung mga molecules na to na hindi basta-basta nakakadaan dun sa semi-permeable membrane. Right, so yeah, they also exhibit specificity. So they're only specific molecules that could be transported by these carriers. So hindi rin naman lahat ng types of molecules or lahat ng types of particles ay kayang dalhin ng mga carrier molecules. Again, they are also kind of like selective on which types of materials they would be bringing into the cell. This illustration right here shows us how carrier molecules would work. So in this illustration, again, the area or the two-layer structure that is seen in blue is yung semi-permeable membrane, which is made up of the phospholipid bilayer. The carrier molecule is this structure right here that is purple in color, and the substance that we would like to move into the cell is these glucose particles right here. Okay, so ang pakinabang daw po ng carrier molecules, since hindi basta-basta nakakadaan yung glucose dito sa phospholipid bilayer, is kailangan dito siya dadaan sa carrier molecule. Now, this carrier molecule is specific. So, hindi lahat ng iba pang substances ay kayang dumaan sa carrier molecule na to. So, this one is for glucose only. So, as we can see right here, glucose could enter, pass through, and then this would also close para ma-regulate or maiwasan yung pagdaan ng iba pang substances. So, this is what carrier molecules would look like. Again, carrier molecules are important in cases of facilitated diffusion. So those were the different methods of passive transport. Let's now talk about active transport. Again, a lot of statements have been said here. Let's just focus on the important ones. So active transport is a carrier-mediated process requiring ATP. So kung kanina nakakita tayo ng kung paano gumagana yung mga carrier molecules, dito naman in active transport, they really require the presence of carrier molecules. But aside from that, they also need to spend ATP. So kung sa passive diffusion, hindi sila gumastos ng ATP molecules, dito they require to spend or use a molecule of ATP. Active transport processes would accumulate necessary substances on one side of the cell, and then this would create concentrations many times greater than those on the outside of the cell. So sa active transport daw po, sinasadya ng cells to accumulate, or yun nga, may mag-iipon siya ng certain substance or certain material doon lang sa looban ng cell in comparison to the amount that is present on the outside of the cell. The most well-known example of active transport is the sodium-potassium pump. All right, so let's look at the statements here. Sodium-potassium pump would move sodium out of the cells and potassium into the cells. All right, so this would result in a higher concentration of sodium outside and a higher concentration of potassium on the inside. So again, sinasadya ito ng mga cells natin. So they're using up energy to store potassium on the inside and to keep sodium on the outside. This illustration right here shows us what the sodium-potassium pump would look like and how does it work. So this layer right here, this blue area right here, is again the phospholipid bilayer. And this structure right here, ito yung mismong sodium-potassium pump. Notice that at the bottom, there is a specialized area where ATP or adenosine triphosphate could attach. So if walang ATP na naka-attach dito sa area na to, this would not work. Alright, so that's what we mean when we say active diffusion. It requires the expenditure of energy, or it requires the usage of adenosine triphosphate. Hindi katulad ng ibang illustrations na nakita natin kanina na hindi naman kailangan ng attachment ng ATP molecule. So as we can see right here, sodium molecules are being pushed or transported to the outside, and then potassium molecules are being allowed to enter. All right, so again, this is the most well-known example of active transport, and this is known as the sodium-potassium pump. Let's now talk about endocytosis. So endocytosis, this is a process that brings materials into the cell using vesicles. All right, ulitin natin yung statement na yun. Endocytosis, they bring materials to the inside, or nagdadala sila ng mga particles papunta sa loob ng cell using vesicles. Or yun nga, yung mga structures that look like bags or that look like sacs that would first need to fuse or magdikit dun sa cell membrane para makapasok yung mga materials na bit-bit nila. There are 3 different methods of endocytosis. First is receptor-mediated endocytosis. So this occurs when a specific substance would bind to the receptor molecule on the outside of the cell before it is transported to the inside of a cell. Let's take for example the idea of having a lock and a key. All right, so kung halimbawa tayo yung particles, tayo yung molecules na kailangan makapasok dun sa cell, kapag sinabing receptor-mediated endocytosis, it's like going inside of your house, wherein you first need a key that would fit the lock of your house. So sa receptor-mediated endocytosis, may structure dun sa labas no cell that would have to fit with the structure of the molecule that would like to go inside. So imagine the structure of the molecule na papasok dun sa cell is your key, and then the structure that is on the surface of the cell is the lock. So those 2 things would first have to match before the substance could get inside of the cell. If it does not match, then it would not work and the substance would not be able to enter the cell. All right, so that's the general idea of receptor-mediated endocytosis. All right, so the receptors on the outside of the cell would have to match with the structure of the molecule that needs to get inside before the molecule is allowed to go inside. Next is phagocytosis. This is a type of endocytosis used when solid particles are ingested. All right, let's compare that to pinocytosis. Pinocytosis, this is what happens when liquid materials would enter the inside of the cell. Okay, so ayun lang po, yun yung keywords na kailangan natin tandaan. When we say phagocytosis, solid materials are being allowed to the inside of the cell. But when we say pinocytosis, liquid materials are being allowed to the inside of the cell. So we could kind of like compare this to eating and drinking. So phagocytosis, imagine this is the way for cells to eat or ingest solid materials, and pinocytosis is a way for the cells to drink or intake liquid materials. This slide shows us how receptor-mediated endocytosis would work. Notice that this is the surface of our cell or the cell membrane. Now, there are structures on the outside known as receptors, and then there are also structures on the outsides which for this one, wala silang specific na molecule na pinangalanan. No? We're just seeing it as molecules that need to be transported to the inside. Notice that the molecules that need to be transported are spherical in shape, and the receptors are kind of like in this half circle kind of arrangement. So their structure would match dahil spherical yung molecule and cup shape yung receptor. So once the molecule that needs to be transported would bind to the receptors, the cell membrane would now change in shape and react and allow these materials to go inside, which, as we can see here in this third segment of the illustration, it has now formed a vesicle containing the molecules on the inside. So eventually, these vesicles would be dissolved and yung mga materials na nasa loob would now be readily available through the inside of the cell. All right, so again, that's how receptor-mediated endocytosis would work. So something has to be, or the structure of something that needs to go inside of the cell needs to match with the receptors on the outside of the cell. Next is the process of exocytosis. So this involves the use of membrane-bound sacs that are referred to as secretory vesicles that accumulate materials for release from the cell. Alright, so basically may lalagyan din daw po or may mga vesicles din daw kung saan yung mga bagay na kailangan ng itapon palabas ng cell would be contained within these vesicles. Alright, so the vesicles would move to the cell membrane, they would fuse, so mag-merge daw po yung structure ng vesicles dun sa structure ng cell membrane, and this would release the materials na nasa loob kanina ng vesicle to the outside of the cell. Examples of exocytosis are the secretion of digestive enzymes, which happens in our digestive tract. Okay, so may mga enzymes daw po na ilalagay muna nila sa container, ilalagay muna nila sa vesicle, and then kung kailan kailangan, these vesicles would merge or fuse to the outside of the cell para yung contents on the inside of the vesicle would now move to the outside of the cell. Again, that's a general example of how exocytosis would work. This slide right here shows us a three-segment or three-step illustration of how exocytosis happens. So there's this vesicle or container. The materials that we need to release are on the inside. So the vesicle would fuse or merge with the cell membrane and then eventually releasing the contents to the outside. This photo right here is an electron micrograph of how this happens. Let's now talk about the general cell structure. So the interior of a cell is composed of a cytoplasm, which again, we've described this as a jelly-like fluid that surrounds and holds the organelles. All right, so basically, yung mga organelles or yung mga maliliit na structure na nasa loob ng cell, they're kind of like floating around thanks to the cytoplasm. So next, the organelles are specialized structures that perform certain functions. So magkakaibang organelles, magkakaiba yung itsura nila, and magkakaiba din yung pakinabang nila. First structure that we would be talking about is the nucleus. So the nucleus is a large organelle that is usually located near the center of the cell. But again, as we progress through this semester, malalaman po natin hindi lahat ng nucleus ay necessarily nasa gitna ng cell. Alright, there may be other cells out there that the nucleus is kind of like on the edge or at the side of the cell. Again, this structure is very much related to their function and the other materials that could be found inside of the cell. Okay, but again, in general, no, for the most part, sa karamihan ng mga cells, the nucleus is located in the middle. But obviously, not all cells have a nucleus right at the center. Depending on the function, some nucleus may be on the sides. Now, the nucleus is bound by a nuclear envelope, which consists of an outer and inner membrane. And these nuclear membranes, they have nuclear pores, wherein materials could pass through in and out of the nucleus. So, same dun sa ideya ng cell membrane, selective din yung pagdaan ng materials dun sa nuclear pores. Pero kasi may mga materials na ginagawa yung nucleus that needs to move to the cytoplasm, which is why we need the presence of nuclear pores so that the materials that was created within the nucleus could move out and be present in the cytoplasm. Within the nucleus are nucleoli, which are diffused bodies with no surrounding membrane, and they are found within the nucleus. The statement is kind of redundant kasi sinimulan niya sa within the nucleus, and then the statement ends with in the nucleus. So it kind of like says the same thing twice, but whatever. There are usually 1 to several nucleoli within the nucleus. So sinasabi po dito na maaaring merong isa or higit pa sa isa, no? Maaring maraming nucleoli sa loob ng iisang nucleus. The subunits of ribosomes, which is also an organelle, is formed within the nucleolus. Okay, so may mga organelles po na usually present sa labas, no, or present dun sa cytoplasm ang nagagawa sa loob ng nucleus or ginagawa ng nucleoli. An example of this is the ribosomes. So these ribosomal components would exit the nucleus through the nuclear pores. So ito na po yung nabanggit natin kanina, no? The function of the nuclear pores. There are things that are being created inside of the nucleus. There are things that they're being created by the nucleoli, and they have to move out of the nucleus eventually. So dun sila dadaan sa nuclear pores. This illustration shows us what a nucleus would look like, the nucleolus inside of it, and these dots on the outside, those are the nuclear pores. These photos right here are electron micrographs showing the actual appearance of these structures. Next are ribosomes. So ribosomes are components that are produced in the nucleolus. So ito na po yung nabanggit natin kanina na material na kayang gawin ng nucleolus. So nasa loob siya ng nucleus pinoform, but eventually it needs to move to the outside. So ribosomes are organelles where proteins are produced. Alright, so ulitin natin, our cells have the capability to produce proteins. Protein production happens within the ribosomes. So the ribosomes may be attached to organelles such as the endoplasmic reticulum. Now, if there are ribosomes out there that are not attached to any organelle, then we would refer to them as free ribosomes. Okay, ulitin po natin. Once the nucleolus produces a ribosome, this would now move to the outside, dadaan ito dun sa nuclear pores. Now, once it's on the outside, it could either attach to the endoplasmic reticulum or it could float around by itself. If it is not attached to the endoplasmic reticulum, then we would call them free ribosomes. Next is the endoplasmic reticulum. The endoplasmic reticulum, or from this point or onward, we could just refer to this as the ER. Okay, so the ER is a series of membranes that could either form sacs or tubules that could extend from the nuclear membrane into the cytoplasm. Alright, so ang endoplasmic reticulum na po, mukha siyang extension ng nuclear membrane. Alright, so a lot of the portions of the ER is still attached or is connected to the nuclear membrane. Later on, may makikita tayong picture, we would understand what we mean when we say it looks like a series or kind of like a group of tubes and sacs. Now, we have what is known as the rough ER. So this is involved in protein synthesis, wherein it has it appears to be rough because a lot of ribosomes are attached to it. Okay, so again, the rough endoplasmic reticulum, this is the site of protein synthesis, which makes sense because ribosomes are attached to the ER or the rough ER, and ribosomes are responsible for creating proteins. Next is the smooth ER or the smooth endoplasmic reticulum. This one does not have any attached ribosomes. Wala daw pong ribosomes na nakadikit sa smooth ER. So since walang ribosomes dito, we won't be able to observe the creation of proteins sa smooth ER. The smooth ER, what happens here is lipid synthesis or the creation of fat molecules, cellular detoxification. So if there are any toxic materials within the cell, ayan po, usually dito sa smooth ER, sila gumagawa ng paraan para ma-detoxify yung mga toxic materials na yun. And sa smooth ER din po, this is where we could store calcium ions if the cell is a skeletal muscle cell. Alright, so again, huwag kakalimutan yung functions ng bawat ER. Again, for rough ER, it functions to be the site of protein synthesis. For the smooth ER, this is a bit difficult to remember kasi marami siyang iba't ibang kayang gawin. So the smooth ER could be a site of lipid synthesis, cellular detoxification, or storage of calcium ions. This illustration right here shows us the appearance of the endoplasmic reticulum. So if we look at this illustration, ito daw po yung area ng nucleus, and then its covering is referred to as the nuclear membrane. So the holes that could be found within the nuclear membrane is referred to as the nuclear pore. And as we've said earlier, the endoplasmic reticulum is actually a continuation or an extension of the nuclear membrane. So as we can see right here, medyo mas malapit sa nucleus yung rough endoplasmic reticulum. And again, it is rough in appearance because a lot of ribosomes are attached to it. And because ribosomes are attached to it, this is the primary site of protein synthesis. Right here, further away, is the smooth endoplasmic reticulum. It is smooth because there are no ribosomes attached to it. And since no ribosomes are attached to it, wala yung organelle na kayang gumawa ng proteins. So dito po, they would be doing other things, which again would include lipid synthesis, cellular detoxification, or kung ito ay isang smooth muscle cell or any type, or actually it's the skeletal muscle cell. Again, if this is a skeletal muscle cell, then they may store calcium ions within this area. Okay, ulitin po natin. Rough endoplasmic reticulum, it's rough because there are ribosomes attached. It is the site of protein synthesis. Smooth endoplasmic reticulum, it's smooth because no ribosomes are attached. And three major functions of the smooth endoplasmic reticulum would be lipid synthesis, cellular detoxification, and storage of calcium ions kung ito ay skeletal muscle. Next is the Golgi apparatus, otherwise referred to as the Golgi complex. So these are stacks of curved membrane-bound sacs. Okay, so again, may hiwalay na picture po to explain that. The purpose of the Golgi apparatus is to collect, modify, and package proteins and lipids that have been produced by the endoplasmic reticulum. So yung mga proteins na gawa ng rough endoplasmic reticulum and yung mga lipids na gawa ng smooth endoplasmic reticulum. And po, this would be collected and packaged in the Golgi apparatus. So this would form vesicles, some of which are secretory, some of them are lysosomes. Okay, so basically, the Golgi apparatus is kind of like a packaging center. Yung mga materials na ginagawa ng cells ay, ayun nga, kokolektahin at ipapackage ng Golgi apparatus so that it could be either released to the outside or maaaring it would be kept in the inside of the cell and would be used for future purposes. Here is an illustration of the Golgi apparatus creating vesicles, and this area right here is an example of exocytosis. So yung vesicle na na-create ng Golgi apparatus, it would move to the outside of the cell, fuse with the cell membrane, and release the contents that it has. Alright, so again, that's the main function of the Golgi apparatus. Shown right here is an electron micrograph of the Golgi apparatus. So this is what it actually looks like within a cell. Next are lysosomes. So lysosomes are membrane-bound vesicles formed from the Golgi apparatus. So sa loob ng mga lysosomes, they have various enzymes, many different enzymes, and usually they function as intracellular digestive enzymes. Let's break down the definition. What do we mean when we say intracellular digestive enzyme? It means that if there is a particle within the cell or nasa looban ng cell, then the lysosomes would work to digest those materials. So again, all of these things are happening on the inside of the cell. Alright, so vesicles are formed by endocytosis, and they may fuse with the lysosomes in order to break down materials. So maaari naman daw po na may mga materials galing sa labas na transport papunta sa loob ng cell na kailangang i-digest or i-break down ng cell. So mangyayari ito with the help of the lysosomes. So one example of this are white blood cells phagocytizing bacteria. So ang mga white blood cells po trabaho nila to ingest or engulf materials that are seen as foreign or that are seen as infectious. Now, just basically materials that should not be within our body. So once these materials have been phagocytized by the white blood cells, anong gagawin? They have to destroy those materials, diba? So they destroy those materials by exposing them to lysosomes. So all those different enzymes that are contained within the lysosomes, they would work to break down the infectious pathogen or foreign material that has been ingested by the white blood cells. This illustration right here shows us how lysosomes work and how phagocytosis could happen. As we can see right here, there are materials on the outside that have come to the inside of the cell through vesicle formation. So itong circle nito, it contains materials that needs to be digested, all right? And then this vesicle right here is the lysosome, which again, it contains many different enzymes that could digest or break down materials. So what would happen here is mag-fuse or mag-merge or magdidikit yung vesicle ng lysosome and yung vesicle ng material na pumasok dun sa loob ng cell. So once they merge or fuse, the materials on this side would be exposed to the enzymes on this side, destroying them. And here is kind of like an end product of that. So this is what it looks like if we're observing the action of lysosomes. Next are peroxisomes. Peroxisomes, these are small membrane-bound vesicles that have the capability to break down fatty acids, amino acids, and hydrogen peroxide. Pero for this discussion, let's focus on hydrogen peroxide. Hydrogen peroxide is a byproduct of fatty acid and amino acid breakdown. So basically, kung dinigest daw po natin yung mga food items that contains fatty acids or amino acids, this could create hydrogen peroxide. Now, excessive amounts of hydrogen peroxide is actually toxic or harmful to our cells. Now, dun na po papasok yung trabaho ng peroxisome. So, the enzymes that are contained within peroxisomes have the capability to break down hydrogen. So when that happens, masisira yung entire structure ng hydrogen peroxide, and this would now lead to creating a new structure na hindi na harmful to the cells. Next is the mitochondria. So everybody knows the mitochondria as the powerhouse of the cell or the ATP center of the cell. All right, so ayan po. As you can see right here, mitochondria is actually a term that is plural. So if we're talking about a single mitochondria, then we should use the term mitochondrion. Okay, so these are small organelles that are responsible for producing large amounts of ATP by an aerobic metabolism. So pag sinabi natin aerobic, it requires the presence of oxygen. So basically, hindi kaya ng mga mitochondria na mag-create ng ATP without the presence of oxygen, right? So the outer membranes have a smooth structure. Meanwhile, the insides have numerous folds, and these folds are referred to as cristae. The material within the inner membrane is the mitochondrial matrix, and it contains enzymes and also mitochondrial DNA. All right, most of our DNA is located in the nucleus, pero nga po, each mitochondria also has their own copy of a DNA, which again is referred to as mitochondrial DNA. So the cells with a large energy requirement have more mitochondria compared to cells that don't. Okay, ulitin po natin, yung mga cells natin na medyo mas magastos sa energy, medyo mas maraming processes na ginagawa na, yun nga kailangan nilang mag-spend ng ATP molecules, they would need to have more mitochondria, which makes sense because again, mitochondria is responsible for creating ATP molecules, for providing energy. So if malakas gumamit ng energy ang isang cell, it makes sense that they need to have more mitochondria. Shown here is an illustration of how how one mitochondrion would look like. Notice the outside is smooth, the inside has many curves and folds, which again is referred to as the cristae. This photo right here shows us an electron micrograph of actual mitochondria within a cell. Next up is the cytoskeleton. The cytoskeleton would give an internal framework to the cell. So yung placement ng internal structures, yung support for the shape of the cell, is mostly thanks to the cytoskeleton. Alright, so it consists of protein structures that support the cell, hold the organelles in place, and help the cell to retain its shape or change in shape if needed. Alright, so there are three different protein structures that could make up the cytoskeleton. So first up are microtubules, microfilaments, and intermediate filaments. First up are microtubules. They are hollow structures formed from protein subunits. So as the name implies, no, they look like tubes. Okay, they are hollow or they have a space on the inside. So microtubules, they have many different roles, including helping support the cytoplasm of cells. So tulad na nabanggit natin kanina, the cytoplasm is a jelly-like fluid that could hold the organelles in place. So now we know na hindi lang naman yung cytoplasm yung nagdadala ng lahat ng ito. Alright, so also helping keep the organelles in place are the microtubules. The microtubules could also assist in cell division, all right? So as the cells would make copies of its DNA and would prepare to kind of like divide to create new cells, and involved din dito ang mga microtubules. They could also form essential components of certain organelles such as cilia and flagella. So the cilia and flagella, these are extensions on the outside of the cell, which we'll talk more about later. Okay? But again, these two are also made up of microtubules. Next up are microfilaments. So microfilaments are small fibrils that are also made up of protein, and they determine cell shape. Right? So ayan po, again, microfilaments, they are fibrils that could determine or could set the shape of the cells. All right? Some microfilaments are also involved in cell movement, and microfilaments in muscle cells are capable, or they would enable the cells to shorten or contract. Medyo mas maintindihan po natin yung concept na ito on muscle cells on why they need to shorten or contract. But again, muscle cells have that capability because of the existence of microfilaments. Next are intermediate filaments. So intermediate filaments, these are also fibrils made out of protein, but they are smaller in comparison to microtubules, but they are larger in comparison to microfilaments. So I guess that's where the name intermediate came from. Now it's kind of like in the middle in terms of size. Again, it is smaller compared to microtubules, but it is larger compared to microfilaments. Right? So they provide mechanical support to the cell. And listed here, or kind of like mentioned here, is a specific area wherein we would find an example of an intermediate filament. So a specific type of intermediate filament is keratin. Keratin is a protein associated with skin cells. So hindi muna natin na-explain yung main functions ng keratin. We would get more into that when we talk about the integumentary system. But ayun po, just know or just understand for now that keratin is actually made up of intermediate filaments. This illustration shows us what it looks like if something is classified as a microfilament, a microtubule, or an intermediate filament. This is an illustration on their placement on the inside of the cell, and then this photo right here is an electron micrograph of what they would actually look like inside of the cell. Up next are centrioles. So the centrosome, this is a specialist area in the cytoplasm that is close to the nucleus where microtubule formation occurs. Alright, so yung microtubules na po, sila ay naka-create dun sa centrosome. So now, one cell would contain two centrioles, and they're normally oriented perpendicular to each other. What does that mean? Perpendicular meaning kung nakaturo sa isang direction yung isa dun sa mga centrioles, the other centriole would have to be kind of like facing or arranged in a different direction. Okay? So each centriole is a small cylindrical organelle composed of microtubules. So ayun po, aside from, or let's kind of like re-explain that idea. Microtubules are created in the area known as centrosome. Tapos doon naman sa centrosome, they have centrioles. So yung mga centrioles po, no, they have the capability to also create microtubules. All right, so yeah. The centriole is involved in the process of mitosis, which we would understand more later. I-discuss din po natin yung steps in mitosis, pero ayun po, wag natin kakalimutan, no, the main organelle that is very important when mitosis happens is the centrioles. This illustration right here shows us what a centriole would look like, and this photo right here is an electron micrograph. I've been saying electron micrograph simula kanina pa. Just to clarify, guys, when we say electron micrograph, we were able to view this image with the help of an electron microscope. Okay, so as we can see right here, we have two centrioles. This one is oriented or facing a direction in this arrangement. So yung isa nakaharap siya sa direction na parang pasalungat, no, or parang nakaharap sa atin, which again is perpendicular, or is described to be perpendicular to the other one. So that's just kind of like how it is for all cells. Okay, so all cells, or almost all cells, okay, would have two centrioles that are arranged facing different directions, or that are perpendicular. So again, this is very important when we get to the process of mitosis or cell division. Next are cilia. So cilia, they project from the surface of some cells. Okay, hindi po lahat ng cells ay merong cilia. So they are responsible for the movement of materials over the top of cells such as mucus. So kung may mga bagay daw po sa ibabaw ng cell na kailangang i-move or kailangang ilipat ng lugar, all right, the cilia are there to move them from one place to another. All right, so again, if we would need to move materials at the top of the cells, or to move them away to a close vicinity, the cilia would be working to move those things. Okay, so the cilia are cylindrical structures that extend from the cell membrane and are composed of microtubules. So ulitin po natin, ang mga cilia ay gawa rin po sa microtubules. Next are flagella. So flagella have a structure that is very similar to cilia, but they are much longer and obviously larger in comparison to cilia. Now, another difference is that for cilia, kung ang pakinabang nila is i-move yung mga bagay na nasa ibabaw nila, ang flagella po, ang pakinabang nila is i-move yung mismong cell. All right, to help the cell to move from one place to another. And an example of this are sperm cells. So sperm cells have one flagellum per cell. Okay, so that one flagellum would help them move from one place to another. It would propel the sperm cells. So that's an example of what a flagella could do. Up next are microvilli. So microvilli are specialized extensions of the cell membrane, which are also supported by microfilaments. So microvilli, they don't actively move in comparison to cilia and flagella. Ulitin po natin, ang pakinabang ng cilia is to move materials that are present on the top of the cell. Ang pakinabang naman ng flagella is to move the cell itself. Okay? Pero ang microvilli, although parehas silang kumbaga extension sa surface ng cell, pare-parehas sila na elongated structures, no? Microvilli doesn't necessarily move anything. Rather, they are simply there to increase the surface area of certain cells, and usually, ang function nila is for absorption. Alright? Again, ulitin natin, if a cell's function is mainly for absorption of materials, we would expect that they have what is known as microvilli. And given here are certain organs kung saan makakahanap tayo ng mga cells that have microvilli. First up is the intestine and the kidney. So sa intestines po, they have the capability to absorb the nutrients that we acquire from the food items that we eat. Sa kidney naman po, they have the capability to reabsorb the water before urine is formed. So, kung halimbawa dehydrated yung person, maaaring i-reabsorb niya muna yung ibang water, and then yung excess water, yun lang yung magiging bahagi ng urine or magiging composition ng urine. So again, these specific organs, one of their main functions is to absorb something. So within their cells or on the surface of their cells, that's where we may be able to find microvilli. On this slide, isimplify na lang po natin kung ano yung sinasabi sa mga statements na to. It says here that the characteristics of a cell is determined by the proteins that it produces. The proteins that it produces is determined by the instruction that is present on its DNA. Okay? Ulitin po natin. Nabanggit natin kanina, di ba, that the ribosomes are the site, or the ribosomes are the organelles responsible for protein production. And then the site of protein production is the rough endoplasmic reticulum. Now, paano malalaman ng cell natin kung anong types of proteins it needs to produce? Again, the types of proteins that a cell produces is dependent on what instructions is indicated in its DNA. So, ayan po, this is otherwise referred to as the genetic information. So, sasabihin ng DNA or naka-encode dun sa DNA na ito yung mga proteins na kailangan niyang i-produce, and ito yung mga characteristics na meron siya in relation to those proteins that they are producing. This slide talks about the DNA. Again, let's just focus on the important statements. The DNA contains the information that directs protein synthesis, or tulad nung nabanggit sa slide kanina, this is the kind of like instructional manual on which proteins needs to be produced. Now, this process is otherwise referred to as gene expression. A DNA molecule consists of nucleotides that are joined together to form two nucleotide strands. Let's further talk about gene expression. So gene expression, this is otherwise referred to as protein synthesis. Okay, so yung proteins na i-create ng isang cell, this is basically the instructions or the genes being expressed by the cell. Alright, so again, in a way, it's kind of like synonymous, no? Pwede nating sabihin na this process is referred to as gene expression, but it's basically protein synthesis, all right? And there are two main steps to protein synthesis, and that is transcription and translation. So transcription po, this involves the copying of DNA into a messenger RNA. And next, the translation naman po, this involves the messenger RNA being used to produce a protein. Okay, so ulitin po natin, sa transcription, kakopyahin yung structure ng DNA para makapag-create ng messenger RNA. So translation, gagamitin na yung messenger RNA para, kumbaga, ito na yung mismong instructions as to which proteins need to be produced. This slide further talks about transcription. So transcription takes place in the nucleus. So the DNA would determine the structure of the messenger RNA through transcription. So as we can see here in the third bullet, during transcription, double strands of the DNA segment would separate. So if you would know, diba, yung DNA structure, it is a double helix, all right? So it's kind of like in a twisted ladder formation. So sa transcription, mag-unravel daw po yun and maghihiwalay yung dalawang portions ng DNA strand, all right? And then after that, it would pair with the nucleotides ng mRNA. So ulitin natin, maghihiwalay yung original structure ng DNA, tapos yung isang strand, it would now connect to the structure of the messenger RNA. On this slide, let's talk about the structure of the DNA in comparison to the messenger RNA. So sa DNA, yung original structure ng DNA, ang base pairs niya is ATCG, or adenine, thymine, cytosine, and guanine. In comparison to the messenger RNA, dito po mapapalitan yung thymine. So we now have AUCG, or adenine, uracil, cytosine, and guanine. Okay, so on this slide, it just lists down the supposed base pairs. So ito yung mga base pairs na present sa DNA, and ito yung mga base pairs na present dun sa messenger RNA. Alright, so thymine could pair up with adenine. Adenine could pair up with uracil. Cytosine and guanine would pair up with each other regardless kung DNA or RNA. Okay, so again, notice kung saan lang po nagbabago. Alright, sa DNA, ang meron siya initially is thymine and adenine. Pero sa RNA, after that, no, ang meron siya is adenine and uracil. So take note of that difference. This illustration right here just shows us how transcription would happen. Alright, so again, ito yung original structure ng DNA. So maghihiwalay yung dalawang strands ng DNA, and then ang magbabind is yung messenger RNA. So didikit yung messenger RNA sa isa dun sa dalawang strands ng original DNA structure. So now we're done with transcription. Let's now proceed to translation. So translation occurs in the cell cytoplasm. All right, i-recap natin. Transcription happens in the nucleus. Translation would now happen in the cytoplasm. So this is after the messenger RNA has exited the nucleus through the nuclear pores. So now the messenger RNA would attach to a ribosome. Aside from that, we would now also have what is known as a tRNA, or a transfer RNA. So as the name implies, it could transport specific amino acids to create a polypeptide chain. Now, if you remember from our discussion on basic biochemistry, amino acids are the building blocks of proteins, di ba po? So basically, ang nangyayari dito, kinukuha ng mga transfer RNA yung mga necessary na amino acids na in-encode ng DNA para, ayun nga, para pagdating dun sa ribosome, no, alam na nito or meron nito ng mga materials needed to create whichever protein has been required or has been instructed to the cell. Let's now proceed to the cell cycle. So during growth and development, the cell division would occur to increase the number of cells. Or pwede rin naman mag-cell cycle sila to replace damaged or dying cells. So cell division involves a cell cycle, and cell cycles have two major phases, the interphase and mitosis. So sa interphase, hindi pa muna nagdi-divide yung cells. Yung time kung saan mag-cell division na ang isang cell is the mitosis. Alright, so let's first discuss the interphase. The interphase is actually divided into three more segments or three other phases. First one is the G1. Okay, so sa G1, the cell would carry out normal metabolic activity. So it's kind of like the expression na business as usual, wherein no significant change is ongoing. Kung ano man yung usual function ng cell, kung ano man yung usual metabolic activity niya, that's what's going to happen in G1. Next is the S phase. So sa S phase, this is when the DNA is replicated. So kumukuha sila or gumagawa na sila ng copy ng original DNA. Next is the G2 phase. This is the time wherein the cell would prepare to divide. Okay, so by the end of the interphase, the cell has two complete sets of genetic material. So dalawang kopya na yung DNA na meron yung original cell, and ready na ito to separate into two different cells. Alright, so this illustration right here just shows us kind of like a visual representation of the cell cycle. This slide, focus lang po tayo sa mahalaga. The human cell, no, contains 23 pairs of chromosomes. So in total, there should be 46 chromosomes. All right, again, for normal, healthy humans, we should have a total of 46 chromosomes. 46, all right, because each cell contains 23 pairs. This slide talks about mitosis. So mitosis is the formation of two daughter cells from a single parent cell. So by the end of this process, dapat daw po merong dalawang bagong cell na na-create galing dun sa original cell. So mitosis is divided into 4 stages: prophase, metaphase, anaphase, and telophase. First stage is the prophase. This is when the chromatin condenses to form visible chromosomes. So dito po, ang main organelle that is involved is the microtubules. So the microtubules would assist in the breaking of the centromere, and then they would move to the opposite sides of the cell. So for this statement, ang pinakamahalagang kailangan lang natin tandaan is kung ano yung organelle responsible or main organelle that is working during the time of prophase, and that is the microtubules. Also, another important thing that is happening here is that the nuclear membrane would dissolve. So yung harang sa pagitan ng nucleus and yung cytoplasm would now be removed or dissolved. Next is the metaphase. So the metaphase, dito po the chromosomes would align near the center of the cell. So the movement of the chromosomes is assisted by the spindle fibers. So again, tandaan po natin dito anong organelle is responsible for these events. So again, in the metaphase, the main organelle that is working is the spindle fibers. Next is the anaphase. So at the beginning of the anaphase, the chromatids would separate, and each chromatid is now called a chromosome. So again, tulad ng sinabi natin kanina, for normal humans, 2 sets of 46 chromosomes would be moved toward the centriole at one of the poles of the cell. So nabanggit natin kanina, di ba? The centrioles are one of the main organelles that is responsible for cell division. So dito po, yung genetic material daw, or the chromosomes, are now moving closer to the centrioles. So at the end of the anaphase, each set of chromosomes have now reached the opposite pole of the cell, or kung nasaan nakalocate yung dalawang magkaibang centrioles. And the cytoplasm would now begin to divide, or maghahati na daw sa dalawa yung cytoplasm nung isang original cell. Next is the telophase. So during the telophase, the chromosomes in each of the daughter cells would become organized to form 2 separate nuclei. Alright, ulitin po natin, sa telophase, nakikreate na yung nuclei nung dalawang magiging bagong cell. So following the telophase, cytoplasm division is also completed, and the 2 separate daughter cells are now produced. So basically, sa telophase po, no, na-organize na o nagkaroon na ng sarili nilang nucleus yung bawat isang cell, and yung mismong cytoplasm ay nahati na rin po sa dalawa. This slide shows us an illustration of all the processes that we've described, and ito naman po, this is what it actually looks like. So these are probably also electron micrographs of what happens during the cell cycle. This slide now talks about tumors. So tumors are the abnormal proliferation of cells, yung hindi normal na patuloy na pagdami ng mga cells. So these are due to the problems occurring in the cell cycle. So kanina, nakita natin yung mga steps ng cell cycles, what happens to them. Alright, so ayun po, tumors are formed because there are certain steps in the cell cycle that is not going normally as expected. So ayun nga, hindi tumitigil yung pagdami nila. So some tumors are considered benign and some are considered malignant or cancerous. So malignant tumors can spread by a process that is known as metastasis. This slide talks about differentiation. So let's just focus on the highlighted areas or the important statements. The process by which cells develop with a specialized structure and function is known as differentiation. So all cells start out as something that's kind of similar or kind of like a progenitor to everything. So what happens is nagkakaroon ng differentiation or nagkakaroon ng parang mga kumbaga is parang in accordance to the instruction that is encoded in the DNA, they would start producing different proteins and they would start having these different characteristics. So kung magsisimula sa isang progenitor ang isang cell, later on, magkakaroon na ito ng iba't ibang versions. So some of them would be brain cells, some of them would be skin cells, some of them would be muscle cells. So that's what we mean when we say differentiation. They start off from one type of cell, and then they would mature into many different types of cells. All right. So during differentiation of a cell, some portions of the DNA are active, but the others are inactive. So may mga certain segments daw ng DNA na ito yung ginagamit for for the creation of instructions or for giving instructions kung anong proteins yung i-create para ma-determine kung anong type of cell siya magiging paglaki niya. So that's kind of like the idea of how differentiation happens. This slide right here just shows us the diversity of cell types or kung ano-anong klase ng cells yung pwede nating ma-observe within the human body. So or the cells in our body could be bone cells, some of them could be nerve cells, or the cells that mainly composes or is mainly found on our brain and spinal cord. Our cells could be muscle cells, and some of them could be blood cells. If you would remember from past discussions, blood cells are also considered as connective tissues, diba? It's a liquid form of connective tissues. This slide talks about apoptosis. Apoptosis is known as the programmed cell death, which is normal and is actually controlled. So, yung apoptosis daw po is, kumbaga, is ito yung timer, no, or clock ng bawat isang cell kung saan dapat titigil na siya sa pagsa-cell divide. So, in the developing fetus, apoptosis removes extra tissues. So, the excess tissues would die dahil hindi na sila kailangan. So, these are usually found between the fingers and toes of the developing fetus. Now, for adults, apoptosis would eliminate excess cells. So, there are organs in our body na meron lang certain number of cells that is needed. So all the others excess or extra cells that would be produced dahil hindi na sila kailangan, they would also be eliminated or they would also die through the process of apoptosis, which again is normal and is actually necessary for controlling the number of cells or the normal number of cells. So again, kapag sinabing programmed cell death, this is referred to as apoptosis. This slide talks about aging. So, ano ang mga effects ng cellular aging sa mismo mga cells natin? Now, this happens because of the existence of a cellular clock, which again, kumbaga ito yung timer ng bawat isang cell. So, kapag natapos na yung timer, ayun nga, mag-proceed na sila dun sa programmed cell death nila, which is otherwise referred to as apoptosis. Next, we also have the presence of death genes or genes that encodes for the time kung kailan dapat ay matatapos na yung buhay ng isang cell. Also, as the cell ages, DNA damage also happens. Now, again, ano bang purpose ng DNA? This is for giving instructions for encoding proteins. Now, when the DNA is damaged, there could also be problems in encoding the proteins, and basically, in the end, it also kind of like influences the general function of the cell. So that's just a normal thing that happens kapag tumatanda na yung mga cells ng isang organism or kapag tumatanda na yung organism in general. In this case, we're talking about humans. So as we age, as we grow older, no, there would be DNA damage. Nasisira yung DNA and some functions may no longer proceed as normal, which again is just a normal thing that happens during aging. That ends our discussion for this chapter. I am Aurel Enriquez. Thank you so much for listening.